Welcome to the wellness talk that's hosted by jointly by the Lehman Alumni Association and the Biology Department. And this talk is aimed at providing informations to mainly our alumni and our future alumni. With this talk, we hope to achieve one particular thing, that is to create and strengthen our Lehman alumni community. And we want to have prepare our current students to help the alumni. And when they graduate, and they will become part of the alumni network. So going to forward together. And so I'm hoping that at some point we will all realize that once you are joined the Lehman College, become part of the Lehman College family, you will always be part of the Lehman family, will always help each other and moving forward together. And so this particular talk is co-organized by Ted Mera, and he is a Lehman alum, and Lawrence, he's the president of Lehman Alumni Association. And I want to shout out a special appreciation to Erica from the Lehman Media Relations for designing this beautiful flyers. This is all her work, and I hope you will enjoy. And and the President Lemon was very interested in our talk, but he has a conflict. He can't join us. We were scheduled to have our provost say a few words to welcome and encourage our students. At some point, he will join us. And we'll also have Dean Mill, Dean of the Natural and Social Science, to encourage us. And later on, they will join us. So I'd like to start by, because there are a lot of alumni in the audience. I don't know when did you leave the Lehman College, but this is a bird's eye view of Lehman College that I actually made, using the Google View 3D. And so since you left, there is, I hope you can see my cursor, there's a new building that was built that's called Science Hall. And it looked, on the ground, it looked like this. This is a beautiful building, and we started using it in 2015. Half the biology department moved into new Science Hall. These are the professors who are currently working in Science Hall from biology department. These are the floors, representing the floors. And so the inside, this is the state-of-art research facility. And we host happy students doing happy research work. And this is when they complete and finish. In 2019, I was very proud to be able to capture the moment when they were walking in the graduation line. And I like to say that I'm the only, this is only biology department is the only program that actually has a graduation picture like this. And so, but that was 2019. And since then, we went through a very challenging period of time. And you've seen this graph so many times, I'm sure. Where we are is here, and we're hopeful. The trend is going down. At some point, we'll go down to zero. But that is where our challenge comes in, and how do we get to zero? At the moment, this is where we are. Of the Bronx, this picture shows the by zip code, the inoculation. And so in our neighborhood, there's we have 46% of people that have had one shot of vaccine. This is according to the New York Health Department data that I dug up last night, and so it's really fresh. And but 40, but if you look at the data more closely, the challenge is really there. Is that from the 18 to 24-year-old population, only 25% or 29%, all the way to 34. And 25 or 29% of the population that have received at least one dose of vaccine. And so that's our challenge. And that's where our students in the virology class took on the challenge and trying to spread the word. Now, they will be talking about vaccines. And so let me set the scene where we're going to talk. And so typically, so if this is the virus, and typically, traditionally, you will get a vaccine by taking the virus, cut them into pieces and inject, and then you get antibody. And then the antibody kills virus and prevents the infection. The new approach is by encoding the mRNA in nanoparticle and then inject, and then generate antibody against the virus. And also in the last few days, you saw hearing news reports or advertisement actually, Regeneron is generating monoclonal antibody that could be used to treat. And that's the effect of getting serum from infected people and to prevent the infection. So that's where we are. And now I'm going to turn over to the first student speaker, Sasha, to present her team's work.
Thank you, Dr. Chang. Just give me a moment. I can pull everything up. All right, are we all good on slide share?
Yep,
Yes.
we can see.
All right, good. So hi, everyone. My name is Sasha Vergas, and I'm a graduating senior here at Lehman College. Today, I'll be presenting on the COVID-19 diagnostics and pathogen surveillance. So at the end of 2019, a novel coronavirus designated as SARS-CoV-2 emerged in the city of Wuhan, China, and caused an outbreak of unusual viral pneumonia. Being highly transmissible, this novel coronavirus disease, also known as COVID-19, as we all know, has spread fast all over the world. So the first objective of this talk is to take a look at the research that has been done to understand how the virus works in order to create ways to fight it. And then we'll move on to understand which and how technical advances are being used to track the spread. So first, to start off, we have an actual image of the virus particle itself. This is a negative stain electron microscopy, which shows the SARS-CoV particle. And as you can see, a characteristic feature of the virus is this club-shaped surface projections around the perimeters of the virus. So the virus particle has different components both in and out that we'll be focusing on today, including the genome, envelope, nucleocapsid membrane, and its proteins. So first, focusing on the components on the interior of the virus, specifically the genome and the N protein. So SARS-CoV-2 has a positive single-stranded RNA genome that is pretty large, having around 30,000 nucleotide bases. And the virus genome provides the necessary genetic information that is required to produce the structural proteins that form the outer layer of the virus particle and the nucleocapsid, or what I'm going to be calling the N protein. So the N protein has two primary functions. One, to hold and protect the RNA genome by tightly coiling around it. And then two, to reduce the host cell's natural defenses against the virus once it enters. Next, moving on to the exterior portion of the particle, the RNA genome is surrounded by an envelope or an outer fatty membrane, which contains the virus proteins. This acts like a bag and holds and protects the RNA. So while envelope viruses don't always have a defined structure, it's typical for coronaviruses to be spherical. And then next, we do have three viral proteins called spike, membrane, and envelope, which are all embedded into the outer membrane. So looking at this zoomed photo of a portion of the exterior of the virus, here we have the large S protein or spike protein, which is essential for the virus to attach and enter uninfected cells, and it's responsible for giving the virus its distinctive crown appearance that is typical of all coronaviruses. Next, we have the E protein, which has several functions that contribute to virus growth and its ability to cause disease. And then lastly, we do have the M protein that gives the virus particle its shape and integrity. It is also thought to play a role in the final stages of infection. So, how does the virus get inside? The S protein mediates the virus's entry into cells by binding to the host receptor called human ACE2 through its receptor binding domain. Once the virus enters the body, it gets proteolytically activated by human proteases, or what we call enzymes. The host protease enzyme used is dependent on the entry point of the virus in the cell, and it's responsible for also activating the virus by cleaving off the S protein. So transmembrane protease serine 2 is used when the virus fuses at the cell surface, and then cysteine cathepsins is used when the virus enters through the endosomes. So now I'll just be showing a short video that shows the entryway of the virus into the cells.
An individual can be infected with SARS-CoV-2 after inhaling airborne viral particles. Upon entering the airways, the virus particles encounter respiratory epithelium cells in the trachea and lungs. The SARS-CoV-2 spike protein recognizes a membrane-bound protein on the surface of the cells known as ACE2, shown here in purple. Once bound to ACE2, the spike protein is unable to open, close, and bend freely, locking it in place. Another cell surface protein called transmembrane protease serine 2, shown here in orange, cuts the spike protein in a specific location. These segments of the spike protein fall away and expose previously hidden parts of the spike. The spike protein can then undergo a series of dramatic conformational changes. During the first stage, the spike protein inserts itself into the cell membrane. Once inserted, the spike protein folds back on itself, pulling the membranes of the cell and virus together so they can fuse. After fusion, the viral RNA is deposited into the host cell, where it hijacks the cell's machinery to produce more virus particles.
All right, so that was just a short video summarizing how the virus actually gets into our bodies and infects our cells. All right, so moving on to the transmission of the virus from person to person, people release respiratory fluids during exhalation in the form of droplets, which carry and transmits the infection. These droplets can be expelled in different sizes and affects how long it remains within the air. When on surfaces, the time for which the virus is active is dependent on the kind of surface it is attached to. In terms of transmission routes, the virus can enter the body in three different ways. The first being inhalation, where one takes in the droplets and or aerosol particles via respiration. The second being deposition of droplets and particles on exposed mucous membranes, such as the mouth, nose, or eyes. And this can come from any kind of splash or spray from an infected individual. And then lastly, you can also get infected by touching mucous membranes, as I said before, your nose, mouth, or eyes, with a hand that has been contaminated by either directly touching the infected respiratory fluid or by touching surfaces that have the virus on it. So now for this portion of the presentation, we'll focus on how scientists have been tracing viruses on a micro and macro scale. So here I'm just going to show a flowchart describing some of the different forms of testing that we'll be talking about today. So this first testing we'll be speaking about is called RT quantitative polymerase chain reaction, or known as the RT-qPCR test. It begins by isolating and converting the viral RNA in the patient's sample into complementary DNA or cDNA. And then next, the cDNA is amplified using DNA polymerase tag. So this testing measures the viral load, and a positive test result indicates that there's a good amount of viral load within the individual, which surpasses the fluorescence threshold. So RT-qPCR is considered the gold standard in detection of virus. This testing is affordable and highly sensitive and specific for viral detection. And these tests are able to be performed in common laboratories such as hospitals or urgent cares. So qPCR is very reliable and fast when you have access to the proper materials, with results taking as little as 4 hours. However, a disadvantage of using this is the use of having primers targeting different viral genome parts, which can be affected by viral RNA sequence variation. As RNA sequences change, a virus is evolving, which can then lead to false negatives. The test results are also dependent on a number of external factors, such as the timing of specimen collection, and then needing trained laboratory technicians. Unfortunately, this testing doesn't capture all of the important knowledge we would like it to, such as the duration for which an individual is contagious or the prevalence of certain haplotypes in a population. Next, we'll be talking about rapid antigen testing. And in this test, samples are taken usually from a nasal swab and put against a paper strip in a plastic casing. If the sample was taken from an individual who has COVID, the antigens present in the sample will bind to antibodies within the paper strip, and in turn, a signal that is visually detectable would show, as we see here in this last portion of the photo. So one benefit of this test would be the turnaround time, which can be in as little as 30 minutes. Due to this short wait time, we're able to run large amount of tests in a short amount of time. And the tests are easily readable, as we saw on the previous slide, due to the visually detectable signal. It also doesn't need any special equipment or skills as compared to the molecular techniques. But unfortunately, this test does have poor sensitivity when the viral load decreases with count values over 30, which is typically the case when patients are still suffering from COVID. This lack of sensitivity leads to the potential of having false negative, which is, in my opinion, a grave consequence in the middle of a pandemic. And then lastly, we're going to move on to the serological tests, specifically detecting antibodies produced by a body in response to the SARS-CoV-2 infection or vaccine. And there are two types of antibody tests, one being LFA, which is a rapid antibody test that visually resembles a pregnancy test, where double line means positive and a single line means negative. The second test here on the right is the ELISA testing, which uses a plate that is coated with the S protein. And the idea behind this is that when you put together the individual's blood and the proteins on the plate, it will bind together and create a colorimetric signal, which is then read by a spectrometer. So some benefits of the serology testing is that it indicates both who had immunity and the number of the population that has been affected previously. More specifically, with the LFA test, the results come fairly quickly, and this test can be taken and conducted anywhere. A con of both of these tests, however, is that there's a possibility of having cross-reactivity resulting in false test results. And for the ELISA, the results take longer, about 5 hours or so, and it needs to be done in a laboratory due to the need of special equipment. Now moving on to the surveillance part of the virus, genomic surveillance such as next generation sequencing allows scientists to decode the SARS-CoV-2 genome to learn more about it by sequencing. So this helps monitor mutations, characteristics, and possible health factors of a new virus variant. And more recently, we specifically have been using amplicon sequencing, where we detect the presence of the SARS-CoV-2 by sequencing the entire viral genome. So variants of concern are viruses that carry mutations that can have worse properties as compared to the wild type virus, and they usually cause more severe infection and make vaccine or treatments have less efficacy. So we have three variants of concern that we'll be talking about today. The first being the 501YV1, which originated in the United Kingdom. And this has increased transmission by 40 to 80% and has also increased mortality rate by about 60%. The next is the 501YV3, which originated in Japan and Brazil. And this reduces neutralization by convalescent and post-vaccination serum and can evade neutralizing antibodies elicited by previous infection. And then lastly, we have the B1427 and the B1429, which originated here in the United States. And this does have a 20% increased transmission. And as we all know, a series of public health measures have been elicited all over the globe in an attempt to reduce the spread. And the CDC advises these following precautions to take. They advise you to cover your nose and mouth with masks. They advise you to wash your hands frequently and use alcohol-based sanitizers or soap. They advise that you cover your nose and mouth when you sneeze to prevent you infecting others. Reduce your outings and social contacts. Keep a distance of at least 2 meters or 6 feet. And most importantly, call the doctor and get immediately tested for any shown symptoms. It's always better to be safe than sorry. And that'll conclude my portion of today's presentation. I want to give a special thanks to all of my team members, Genesis Escalera, Philip O'Leary, Jelena Bender, and Nolan Osai. And a special thank you to Professor Rohit Jangra, who's been teaching us all semester. And correct me if I'm wrong, Lawrence, are we taking questions now or after all three?
The questions will be, so please hold all the questions and to the end of the three talks, and then we'll answer all questions. And I'd like to interrupt, quick interruption. I'd like to introduce Dean Pan Mill for to say a few encouraging words for us.
Hi, good morning, everybody. A few encouraging words, huh? This beautiful Saturday morning. I guess I'd like to know the audience I'm speaking to, and I think almost all of you are muted. And so I'm going to ask maybe everybody turn off your mute if you don't want to be on camera. And I'm going to ask, how many of you are alumni? Say yes.
Yes.
One, come on.
Yes, 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 yes.
All right, there should be a lot of alumni here. And how many of you are students at Lehman?
Yes.
Yeah.
Yes.
Yeah.
Yes.
Congratulations, all of you. I have to say, the first talk, I didn't think I was listening to a student. I thought I was listening to a professional talk. Actually, just kidding. I expect that quality from the students. You guys are so good. So I just want to say welcome. I think what you're doing is awesome. Welcome to the alums, and good luck to everybody. I look forward to the next talks.
Thank you.
Thank you.
So we move on to the second talk, which is presented by Anita. Please take over.
Yes. Let me do this. Hello, everybody. My name is Venita. I'm majoring in biology, brain sciences. Today, I'm going to talk about vaccines and COVID-19 pandemic. So today's agenda is what is a vaccine, the common virus vaccines, what is an mRNA vaccine, and then I'm going to specifically talk about Moderna, Pfizer, Johnson & Johnson vaccines. So what is a vaccine? Vaccine is a substance that helps the body to activate the immune system to help fight against the disease. It can elicit the innate and adaptive immunity system. Now, the innate immunity system is the first line of defense, and it is non-specific, and it cannot induce immune memory. However, it can send signals and activate the adaptive immunity system, which indeed it is very specific, and it does induce immune memory. The adaptive immunity system consists of humoral response, which is the antibody-based mediated by B cells, and also the cellular response, which actually happens within the infected cells and is mediated by T lymphocytes. So before the vaccines, smallpox was an infectious disease caused by variola virus and had a high mortality rate. At that time, violation was practiced to help eradicate the disease. This was done by taking a small scrap of the disease and inoculate it into a non-infected patient. Now, this was dangerous because the patient could die from the procedure and the mild form of the disease could spread and cause an epidemic. So when did the idea of the vaccine come to life? The idea of the vaccine was introduced in 1796 by a British physician named Edward Jenner. What he observed was that in the milkmaids that were infected by the cowpox virus, they were protected by the smallpox. Since cowpox virus causes a very mild disease in humans, he used that to vaccinate those that were infected by the smallpox. So the vaccines can help, eradicate the viruses. And here's the summary in case of like measles. As you see here, the introduction of vaccination in 1968, helped to eradicate this virus. And you can see here, like the cases are decreasing until finally, like around this time, 1998, it was completely eradicated. And another example is polio and influenza type B virus. So let's move on to the common virus vaccines. One of them is attenuated vaccines, which contains a small amounts of the weakened virus that triggers the immune system to fight it up without actually causing sickness. This vaccine is often used to prevent viruses like chickenpox, smallpox, and measles. The protein subunit vaccines contain harmless pieces of the virus, like proteins. They do not have any live component of the viral particle, and this method is used when creating hepatitis B vaccine. Inactivated virus vaccine. Now, this vaccine is also used for COVID-19 virus. And the vaccine is the Sinovac vaccine, which contains the killed COVID-19 virus. So basically, like the virus, as you see here, is the nucleic acid is being destroyed, so the virus cannot replicate in our body. However, the antigens are intact because in order for our immune system to recognize and start fighting and we can build immunity. Now, the disadvantage of this is that it requires the booster shots to maintain the immunity. The viral vector vaccine was introduced in 1972 when Jackson et al. Created the recombinant DNA from SV40 virus by genetic engineering. A modified version of the virus is used as a vector to deliver genetic code for the antigen. Major vectors used are retrovirus, vaccinavirus, adenovirus, and coronavirus. The example of this vaccine are Johnson & Johnson vaccines and AstraZeneca, which I'm going to talk about in the next slides. Now, disadvantage of this vaccine might be that the host can have immunity to the viral vector due to prior exposure. And the mRNA vaccine, which introduces an mRNA sequence that codes for a specific antigen. Now, in the case of COVID-19, it will code for the spike protein. Oh, sorry. What is mRNA and what are the advantages of using mRNA? Now, we utilize mRNA because our body naturally makes it to form proteins, and this is called the central dogma. So basically, from DNA is being transcribed into mRNA, and then mRNA is going to be translated into the proteins. And here's the example of a cell, a nucleus, and how the DNA is being transcribed into mRNA, and then it goes through all these processes through translation to form proteins. Now, the advantages of using mRNA is that, first of all, like it can be controlled. It is non-infecting and non-integrating, meaning that mRNA will not integrate into our genome. Also, it has the potential, like to be for a rapid manufacturing, right? And then also an important aspect of it is that the mRNA can be degraded afterwards by just normal cellular processes. So if it has all these advantages, why hasn't been used before? Well, scientists had to overcome some challenges in terms of making the mRNA vaccine because mRNA has, in order for the mRNA, like to be stabilized and to be able to translate efficiently, it has to have that 5' cap and the 3' tail, which stabilize the mRNA and increases the translation. So scientists had to find a way to add this 5' prime cap and 3' tail. But not only this, they had to like create and come up with the right lipid nano particle to kind of protect this mRNA from degrading once into the cell. Not only to help, like unprotect it, but also to help the mRNA to get into the cell, to fuse into the cell. So mRNA has not been used before until now for the COVID-19. However, it has been tested before, and it was studied before for the different viruses such as influenza, rabies, Zika, and cytomegalovirus. As you see here in this table, for the Zika virus, there were like clinical and preclinical trials going on trying to see, like having some vaccine candidates for that. And as you see here, mRNA was one of them. And it has been shown to be, like very promising and successful, especially after they they made some nucleoside modification, and also after they formed this lipid nanoparticle. However, there were like setbacks because of the lack of funding. And here we are today at the mRNA vaccine, Pfizer and Moderna, that are being widely used in United States. And you can see here the lipid nanoparticle, which I talked about before, which helps the mRNA to get into the cell and, like it protects from being degraded from the enzymes in the cell. And then we have the mRNA, which encodes the version of the spike protein with 2' modification. So once in the cell, it's going to be translated, it's going to form these spike proteins, which once they leave the cell, like they're going to induce or elicit both the humoral and the cellular response. Now, Pfizer and Moderna have been shown to be 91.5% efficient in preventing COVID-19 illness. They both require 2 shots. However, Moderna is less temperature sensitive, so it can, like it's easier to store and distribute. In both Pfizer and Moderna, there were some cases of allergic reaction. However, these cases were rare. And then the viral vector vaccine, in this case, is the Johnson & Johnson vaccine and AstraZeneca vaccine, which uses the adenovirus viral vector. So basically, as I said before, it can be a disadvantage, can be that the host can have immune to the viral vector, and to overcome this challenge, the scientists use the ADV6 adenovirus serotype. So in the DNA, there is the sequence that codes for the spike protein, and then again, it's going to, in the nucleus, is going to be transcribed, and then the mRNA will be translated, form the spike proteins, and then again induce both humoral and cellular immune response. Because it's the DNA, DNA is more stable, therefore the Johnson & Johnson vaccine is more stable, so it does not need ultra cold storage. It has been shown to be 85% effective in preventing hospitalization and death, and it only requires 1 shot. Now, for this vaccine, there are some cases reported of blood clots, uh, this case these cases were very interesting because all these patients, they had very low level of platelets, which means usually, normally, they actually would have trouble to form these blood clots. So this is considered a rare phenomenon. They do not know, like the absolute answer yet. However, they're still looking into it, like why this is happening. Anyway, the risk of getting the blood clots so far is 1 in a million. Now let's talk about the risks and side effects getting the vaccine versus COVID-19. Now, I hope by now you understand that getting the vaccine, it doesn't mean you're being infected with the COVID-19, right? So you have no risk of death, as in the case of getting COVID, and you have no risk of like long-term effects, right? So if you get the vaccine, the side effects will be like headache, like fatigue, muscle aches, but no long-term effects. Versus if you get the COVID-19, you will have long-term effects because like you're being infected and it's affecting your cells and your organs, and of course the extreme like case is that you might die from COVID-19. So I hope by all this research information now you can make an educated decision to get vaccinated. And I want to thank my group, Michelle Ramirez, Ciara Williams, and Lucille Gill for all their contribution. And of course, I want to thank Professor Ethan for all the feedback and the help. And thank you all for participation. And these are my references. Now I will pass the talk to Brian. Let me stop sharing. Okay. So Brian, whenever you're ready.
Good, right? You can see?
Yes.
So my name is Brian Spikovic. My group members were Daniela Basave, I'm MC, Joel Nieves, and Elizabeth Salazar. And what we worked on is we're more about the safety. We're also going to speak about vaccines, safety and efficacy. My project is going to have a lot of graphs, and we're going to be showing graphs, a lot of graphs. So first question is, how do we get the vaccine so fast, right? That's a lot of things on everyone's mind is they're worried about it because we got the vaccine so fast. So the first thing is in America, there's this thing called the emergency use authorization, where we can speed up this process, right? So the Secretary of the Human and Health Services has to declare an emergency based on data from a report from one of these other secretaries, right? Department of Defense or Homeland Security, hospitals, one of these other secretaries needs to have a report with actual data saying this is an issue, we need help on this, and send it to the Human Health Services, which he's the one who sends it to the FDA, and it still gets issued emergency use authorization. It's still all the normal FDA processes. So what's the difference between regular FDA approval and the emergency use authorization? So on the right here, we have the FDA approval. So it's developed, it gets tested, 3 clinical trials, a lot of clinical trials, yada, and then it's approved, it's produced, and it's distributed, right? It's a slow chain. It takes a while. Normally this could take a couple of years, whereas this vaccine, right, 8 months or so, or whatever. On the emergency use authorization, the production and the testing is happening at the same time. So they're kind of banking on these things working as they're testing it. They're making lots and lots and lots of them. And then it still has to go through approval like a regular vaccine. It's not like it's not getting approved by the FDA. And then it gets distributed, right? Quality control, just like normal. And it starts to roll. Oops, sorry. So what's a normal clinical trial, right? So normally there's 4 main phases, right? Preclinical, which is things like cell cultures, animal models, computer models, things like that. And there's usually a first phase, which is a small number of people, 10, 15, sometimes case studies, 3 people. Phase 2, which is larger, a couple hundred, things like that. And then phase 3, which is usually thousands, right? You want to represent an actual large group of people. So for mine, I'm mostly be focusing on Pfizer. Pfizer actually combined a lot of the clinical stages. So it's kind of clinical 1 and 2. So for their preclinical, they use rhesus monkeys. And what they did was they gave them the normal doses like they would have given us, the 2 doses separated 21 days apart. And then 21 days or 55 days after that, they exposed them to SARS-CoV-2. And then they searched their lungs and their mouth and other areas for viral DNA. So this here is a graph of the viral RNA versus how long since they were infected. So the blue is the vaccinated ones and the control is the gray ones. And you see the blue had no viral RNA, whereas a couple of the controls were actually infected. And this is in their lungs. So this is found in their lungs. So this is showing that it's effective against stopping infection. So that was the first thing in monkeys. That was the first thing they tested. So then they started testing in people, right? These 2 graphs are showing your CD8 T cells and your CD4 T cells, which are your CD8s are kind of the mediators in between your 2 types of immunity. And there's lots of other different types of T cells and stuff. So people always think that antibodies are like your only immunity, but it's not, right? There's a lot of, your immune system is very complex. There's a lot of other cells. So all this is showing is that the low dose and the high dose are eliciting an immune response, right? So the first one's placebo. Both of the charts are showing the same thing. The first one is the placebo. There's nothing, right? And then if you inject them with a low dose or a high dose, doesn't even really matter. They're going to have some sort of immune response. You know, interferons and CD8 T cells, all those things are going to start moving. So then the next question is, do we do 1 dose? Do we do a low dose, high dose? What are we going to do dosage wise? So this is a really good graph for showing this. This is also Pfizer, same study. We're going to compare these first 2 graphs and then the second 2 graphs. So the first one is a low dose and then a placebo, and then the next one is a low dose and a low dose, right? So they got a dose and then they didn't, and then the second one they got 2 doses. So if you look at the days, right? Sorry, the last 2, they sort of flatten out in the low dose and placebo, right? They don't get any more protection. They don't get any, it's plateauing. But then if you look at the double low dose, it continues to rise, right? And then the same thing with the high dose. First, you have a high dose in the placebo, and then a high dose and a high dose. In the placebo, it sort of plateaus out. There's no more increase. And then in the double high dose, it keeps steadily increasing. So ultimately, they went with the double high dose. This is just another picture kind of showing the same thing, right? That you get, this is your region binding domain antibodies, and they took a blood sample and measured the levels. And on days 21, which is about where you get the second shot, it's going to increase again. This is what we want. And this is 3 different levels of strength of the vaccine. So next thing is side effects, right? People always are worried about side effects, but there's actually not really many side effects, honestly. So the biggest thing would be probably your fever, right? But it was only about 17% of 18 to 15-year-olds or 55-year-olds, sorry, had a fever, and fatigue and chills. And you have all these other things, but there's no severe side effects. There's actually, I took off the key, but there was a red section that was severe, and there was actually not a single person in the study. And this study was about it was like 30,000 people or something like that. I forget the exact number, but there was not a single person with any severe side effects, right? And even gray, there was no really gray. There's a little bit of gray. Gray's like extreme, blue and green were medium. So there really isn't even that many side effects So this is actually a very cool data I took from the English Journal of Medicine. It released about a month ago a study about Israel's population. It was like something like 400,000 people they looked at and matched vaccinated people to non-vaccinated people and looked at the effectiveness of that. So this chart is showing the first one it's a time, right? First one is 14 to 20 after the first dose. Second point is 21 to 27 after the first dose. And then the third one is a week after your second dose. So all these different things, right? Severe illness, hospitalization, symptomatic illness, and just a regular infection. You want a higher number. More effectiveness means we're having less of these things. So after 2 doses, all of them go up to about 90%, right? Even severe disease is the same as these documented infections. Whereas after 1 dose, it's less than 50%, right? It's only 46 or something like that. So this is just another graph showing how effective the 2 doses is versus the 1 dose. So the other thing that people are worried about is mutations, right? So this is another good study. They took blood samples from 3 groups of people. One were negative people that didn't have any exposure or vaccination. One through a group of people that were vaccinated, and then a group of people that is convalescent, which is people that were previously exposed to an infection. And then it compared those 3. So if you look at the negative, right, there's no inhibition, right? It's really low compared to the others. And then if you look at the 3 levels of antibodies, low, medium, and high, way more than negative. But then if you look at vaccinated, it's even more so than these other naturally infected people. So vaccination could even give you a better protection than natural immunity, right? And then the other thing this graph is showing is that there's mutations, right? And this is the variety that we've been getting, USA, blah, blah, blah. And this is a mutation that can be found on it. And it's showing that even these 2 mutations, it's still effective, right? It's not like these numbers, some of them are a little different, but they're all around the same range. So it'll be effective against this mutation too. So this is sort of showing that too. This is another mutation issue. So this is a neutralization of all of these different variants, right? And the wild type is what we're going to see. And if you look at the graph, it's all around the same level for all of these mutations, right? So why is that, right? So this is a computer model of the S protein. And if you look at all these tiny little mutations, that's all they are, one tiny little mutation. So all of these little antibodies can still get to the other epitopes and stuff. They're just because there's one mutation doesn't mean that the antibodies aren't effective, right? There's still a million other ways. It's not like the whole S protein is this giant different blob, right? It's just one tiny little mutation. So this is another good study that actually just came out like 2-3 weeks ago. This was Qatar, I believe it was, but it was their same thing, vaccinated population. So in their country, they had 2 variants mostly, the B1351 and the B117. It was 50% and 44.5. So that's 95% were these 2 variants. So the graph on the left is just effectiveness from infection, right? So it had about 90% for the first variant and about 75 for the second variant. But then if you look at the level of disease that they got, these 2 variants were actually protected completely from any severe disease. Other red was actually other. So I don't even remember which variants were off the top of my head, but it was other versions of the virus that wasn't the majority of the population that actually were causing the severe disease. So now I want to talk about price a little bit, just to put in perspective how little it costs, right? So I made this little image. Each of these rectangles is $20 billion, right? This down here on the bottom left is the Medicare budget, $644 billion. So the CDC actually has this really cool chart you can look at, and it'll tell you the price of all of the vaccines that you can find in America. It's crazy. It's a huge, giant document. So I made just a little comparison. To vaccinate every single person in America with hepatitis B vaccine at $25, it's this little tiny triangle, square. MMR, $49, it's this little rectangle. Influenza, $15, it's this little rectangle. And then to even less, I made up a fake number, right? Say $20, the COVID vaccine. It's cheaper than some. It was like an average. To vaccinate every single person on Medicare, all of this money, it's only this tiny little rectangle. It's only $1.2 billion, which I just want to show that it's not that the money is not there. We just have to prioritize. Last question is, should we be getting as many people vaccinated, right? So there's this thing called herd immunity. I'm sure you've heard about it. People are always talking about it. So what it is there's a level of, so we're gonna look at this chart. There's a really good chart. Blue is susceptible individuals, red is infected individuals, and yellow is resistant individuals. So as the level of resistant individuals decreases, right, the level of people that were vaccinated, the level of susceptible people is going to decrease, and the level of infection is going to go to zero, right? There's this kind of crossing point, this perfect spot where they all meet and then they finally all go to zero. Well, I mean, resistant would go to 100. So little image right here. So naive population. This is a population that didn't get any vaccines or anything like that, right? The little red guy goes outside, he infects 4 people. Those people infect another 4 people. And then as soon as you look at it, boom, the whole population's infected. And there's only one guy that wasn't infected. Now, if you look at a herd immunity case, in this model, it was using 70%. This red guy goes to infect people. He can't infect these because they're resistant. He infects 1. This one guy goes to infect others. No one gets infected because they're all resistant. So all of these blue people that didn't have a vaccine, this is your grandma that can't get vaccinated because she's really old and sick, or someone who has an immune disorder, or someone who's on medication, whatever reason, specifics don't really matter. People that can't get vaccinated for whatever reason are protected by the herd immunity because the other people are vaccinated and aren't spreading, right? It can't go 1, 2, 3, 4, 5 if it stops at 2, right? So for COVID-19, we don't really know the exact rate, but I found a model on a paper, and it said about 50 to 80% of the population needs to be vaccinated. It's always a range. There's never like a perfect exact number. It's based off the R naught, right? Like how many people one person will infect. But all these other ones, had similar numbers. Rubella was, 60 to 80. Polo, same thing, 60 to 80. But they're all ranges, so we don't really know yet 100% for COVID. And last thing is, I just want to say that it's you want to go get your vaccine, right? Not to preach, but the side effects of getting a COVID infection are going to outweigh almost any side effect you're going to have from getting the vaccine. You know what I mean? And that's all I want to say. Thank you to my group coordinator, Dr. Catalina Flores. She gave me a lot of feedback when helping me for the project. And I think we're going to open for questions.
Thank you, Sasha, Vanita, and Brian for the very, very excellent and professional talk. And I hope everyone enjoyed it. So now I'd like to introduce the 3 professors that guided these 3 groups of study, and they will join the 3 students to form a panel to answer questions. And so, so these 3 professors are true virologists, current virologists working in Albert Einstein Medical School. And so they took out their busy time to teach the virology class for us and mentoring our students. And I just wanted to say that as a chair of biology department, we really, really appreciate, and for our students, thank you. So Professor Chankra, Professor Londa Mack, and Professor Florence. So I'd like to invite questions. You can put in the chat. Lawrence will help me as well. So we'll monitor the chat, and so to answer questions as they come in.
Excellent. We had one question earlier, but a lot of people kind of helped out. The question was for Sasha, and that was, is the qPCR is used to test COVID-19? And I think there was a lot of answers on that already, but that was the first question coming in.
Yeah, so just to touch back on that, like everybody said, it is used, is actually used as the primary test because it is considered the gold standard, and it is the most reliable testing that we have for COVID right now.
Excellent. Excellent. And there's another
question on why do you still get COVID when you get the vaccine?
Since I talked about vaccines, I can take that one.
Yes, please.
All right. So as I explained you guys, getting the vaccine, uh, what's happening basically, it's helping your immune system to recognize the spike protein that is in the virus. So it builds, as I explained to you how the immune system works, it builds memory, immune memory, right? So getting the vaccine, it doesn't mean that you will not get the COVID. You might get the COVID. However, your immune system is already prepared for that. So it's going to right away start fighting it, and you will not get, like so sick, and you will not get long-term effects as if you would have without being vaccinated. You understand? This is one of the main reasons. And then if you go later to even what I said, even what Brian said, not only it will like protect you, but it will later on basically help to eradicate this virus at all if we all get vaccinated. Is that clear?
Can I add something to that? So I think we really have to differentiate two things: infection and disease. Getting infection does not mean you are getting disease. As we know, and this is one of the arguments that a lot of people are using, that why should I get the vaccine when, most of us get the virus and they are fine. The point here is the vaccine is protection against the serious disease. Does not mean that everyone who is getting the vaccine do not get infected. There are some cases of infection, but in terms of protection from serious disease, hospitalization, needing oxygen, there is still, I would say, 90% or more protection. So that's the key statistic here.
And if I can just jump in and answer the second part of the question, which is why do they have to provide awards such as food metro cards and etc. Coming from the public, I work in the public health field. So the reason we provide, I know when we were trying to get home health aides to get vaccinated, it's because we were providing them with $100 if you come get vaccinated. And the reason we did so is because, getting vaccinated, as we all know, it takes time to out your day to go get vaccinated, to go get vaccinated. And, you got to get out of work, and sometimes you're working from 8 to 5, or you're in a pandemic and, you need food. So how do we get people to come and get vaccinated so we can return to normal when they don't have money to get MetroCards, when they don't have food at home to cook? Why not provide these things as a gift or like an incentive to come and get vaccinated so we can get rid of this pandemic and this virus?
Thank you. So there's another question now. Why do we need 2 doses in 3 to 4 weeks' time?
So it's not so much that it's like it has to be perfect 3 and 4, but that's just what we studied and we know is effective. So we want to use what we studied, right? And the Pfizer and the Moderna were the 2 weeks apart, or Moderna was like 30 days. Pfizer was like 21. But the whole dosage is so you have increased protection. There was that one graph I showed of the antibody levels, and they sort of plateaued, but then you needed that second dose to increase them even more. And who knows, that last little bit of protection might be what stops you from getting severe illness.
Thank you. So why mRNA? There's a question now. Why mRNA, not a weakened form of the virus like all other vaccine?
So there's both, right? The like Sasha was talking about, the Johnson and Johnson is the one that is like a weakened form, the vector virus, but the Pfizer and Moderna are the mRNA vaccines.
Well, Okay, yeah. So since I talked about there are different type of the vaccines, as I talked about in my presentation, right? And they both have their benefits and their disadvantages. And so using like mRNA vaccine, as I explained, is like it has so many advantages because there's no live virus in the vaccine. You just have that mRNA that is sequencing for the spike protein and is helping you build immunity. And you're not being infected. The mRNA cannot insert into your genome, and it basically your cell is going to degrade it later. You know, so like this is why now it has been shown, like to be very even effective and, like a very good choice. And I read a question, why wasn't this mentioned before or used before? I explained that even though the mRNA vaccine was not approved or used before, it doesn't mean that it came out of nowhere, because the mRNA vaccine was being tested, as I explained, for different viruses such as influenza, like Zika virus and different type of viruses. However, they didn't get to this point. There were lack of funding, and then there were all these challenges the scientists had to overcome in terms of making this mRNA stable enough to actually get translated and to actually, boost our immune system. So it has been, like studied before and tested before. It came to this point where this pandemic, like came, and then everybody started to look into it. And, like, thanks God, we're at this point that now mRNA, like is being used for all the advantages that I said about.
Thank you. So, how do you? There was a question to all of you. How do you think How do you think we can better inform the population on different vaccine? And that's what we're doing today. And all three of you did a wonderful job. And the follow-up question is, this presentation allows us to know the second dose is a inactive virus and weakened immune system, weakened virus like the Pfizer or Moderna. Can we explain the uses of differences to encourage others to get vaccinated? And I think, yes, I'm hoping that we will somehow get our three talks and post them online so that they become available for all others to follow up. And so there's a question on the the vaccine that we have and the Indian variants. Go ahead.
Can I just add one thing onto that quick?
Yes.
For me personally, I know if there was one clear message, right, from the health officials that was supported by everything, even if it wasn't, I feel like a big issue is you hear conflicting things everywhere. There needs to be, I think, a big thing about helping inform everyone is, you need a very clear message. This is safe. There might be tiny little effects, but it's nothing severe. And that was, I just want to say that.
Well,
Sorry.
We have 147 participants, and this is the biggest ever crowd that we ever had. So and everyone's still here. So I'm starting to lose track of questions that flies in.
I'll grab
So if I didn't get to your question, I apologize. Exactly. RNA. So there's a question about RNA vaccine as the future for the new vaccine. Anyone want to comment? And I think we all agree how fast people can turn around.
I can take that because,
Mm-hmm.
Yeah, so I think the thing, and this question has come up, right? Why you didn't go the inactivated virus route and why did we choose mRNA? One advantage of mRNA is its synthesis and its testing and everything can be done really fast. And this is being presented as the future of the vaccines for the right reasons because we can respond much faster. So the perfect example is the Pfizer and Moderna vaccines that we are talking about, because from the discovery of the virus to getting to the vaccine and approval, it was less than 1 year. It seems short period, but we did not, again, just to emphasize, we did not compromise any of the safety requirements. The safety requirements and everything was just tested as we would do normally, but manufacturing and everything else is much easier with an mRNA vaccine. And that's why this is being presented as one of the future of vaccine for multiple vaccines, as Benita was also talking about, like Zika, influenza. So I imagine many more of the vaccines taking the RNA route. Doesn't mean that everything will be that way, but this will definitely be explored much more in the future.
So there's a question.
Oh, sorry, Dr. Chang. I was just going to say that there's some really interesting conversation in the chat. Some people were asking about if you've had COVID-19, should you get vaccinated? And someone just gave an anecdote that they know someone who experienced more intense symptoms after vaccination than she did when she first had COVID. So I think I'll let the students take it away, and then we could jump in if necessary.
Personally, I think that if you had COVID-19 and then you're thinking, oh, should you get the vaccine or not? I think that this must be discussed with your doctor because they have, like all your information of your health and everything. And like because we so far like we don't know like exactly how long the antibodies of the virus will last. It was approximately 1 year, but like there's still research going on. So like it all depends. You have, I believe, except all the information that we're telling you, I think you still have to talk with your doctor and then decide. That's my suggestion.
Yeah, so sometimes people who are vaccinated actually have higher levels of immunization. So yeah, definitely talk to your doctor, but I would say yeah.
Yeah, and then in reference to the anecdote of someone that had more intense symptoms after the vaccine, that could have definitely just been the immune system actually responding to encountering the viral antigen once again. So the fever, the chills that the person probably experienced after vaccination, it wasn't actually due to the virus, or I mean, it was an mRNA vaccine, so there is no virus, but it was just because the body was protecting itself, and it was the immune response behaving as it should.
Yeah, people want to think that side effects are like that necessarily, but it's not bad, but it's not really always a bad thing. Your immune, it shows your immune system is doing something.
And just to add to the comment about the longevity of antibody response, as Vanessa was saying, and there is, a little bit of information, depending on, when you got the information. So there have been studies showing where you lose antibodies in 3 months, 6 months, 4 months. That is actually not, well, you do see a decrease in antibody after a certain time, but that does not mean you are not protected anymore. So now we have been almost a year into this thing, and we know that up to a year, those antibodies persist, and people are way more likely to be protected. The people who are carrying those antibodies are way more protected than someone who does not have any antibodies at all. So it's not that those antibodies or your immunity is going away in 3 months, 6 months, or 9 months. Like we have data right now to show that people are protected, and as time goes, we will have more data. We cannot have data, of course, are the antibodies protecting for 2 years, because it's not been 2 years. So if you are asking that sort of data, of course we cannot generate that.
Here's another question that had been circulating a lot. I've heard it about too. Is that this question about regarding to fertility? How can we know this vaccine won't affect or leave a long-term effect on women or men fertility? I guess that's the general question. Anyone want to take it on?
Yeah, I think so. One plug that I can do, I think I don't know how many of you watch John Oliver. He's a comedian on HBO. He puts out these videos on YouTube. You should really watch one of his shows, his recent one about vaccines, specifically COVID vaccines. But coming back to the question, the fertility question is really, it has been raised by some there is no scientific basis for that. If you think about, there is one idea that, well, the virus is making protein that is similar to what is made by placenta. Well, placenta is made by the baby when conception happens. That is not something that exists in women or men who haven't, the woman who hasn't conceived yet. So how can, this vaccine really affect that? Two, if you actually get COVID, you are way more, you will be exposed to way more of these antigen, even if you are worried about that, and even if you believe that that's the basis for your reasoning, you are way more likely to see probably 1,000 times way more antigen than what you will see with the vaccine as compared to an actual infection. And we have so much data now, millions of people got the vaccine, and thousands of them have gotten pregnant, and they are carrying their babies so far. We really do not have any direct evidence to show that any of these vaccines are having any negative effect on fertility over the period. Now, if someone asks how do you know 30 years from now there won't be any effect? Well, the direct answer to that is we do not have that data, but we have a lot of data from a lot of different vaccines, because when measles vaccine came out, people were asking the same questions. How do I know it's not making us important? When polio vaccine was given I remember in high school, I was going home to home in India, giving kids polio vaccine, and there were some people refusing to take it. It's like, because my child will be important. I mean, India's population is an embodiment, and two, it's like there is no importance being caused by the polio vaccine, because we're still growing, right? So yeah, I mean, these are important questions to ask, but you really have to go behind the reasoning, like what is the basis of those. And if you just scratch the surface a little bit, and all of you especially, many of you have taken biology, and if you just scratch a little bit, go beyond what the headlines are, read the article, and all of us are smart enough to really answer these questions for ourselves.
Can I just add something?
Yes, please.
So I work in the hospital, I do ultrasound, and during this time, during the pandemic, I've scanned a lot of patients who were pregnant and they give birth and everything, that's just some real life thing. And also to say that you are infertile or something, it takes up to 1 year. So it's like a standard thing that the doctor cannot say you're infertile unless it's been like 1 year of you trying. So there's some, preconization and stuff that tells you. So it's really, it's not something that we can, say it now because it's been little over 1 year. So it's really is a process, but also there've been plenty of, people that, got pregnant, gave birth, everything looks fine. However, they're still working on this, we'll have data more. But again, you need at least 1 year for the doctor to tell you if you're infertile or not.
I think there's this question I really like in the chat. This probably go to Brian, but Joanna asks, why was this vaccine created so fast, but other things don't have cures? And I think Brian explained that very nicely with his graph. So
Yeah, honestly, the big thing would be money. If I had to give you an answer, right? Everything in the world is driven by money, right? So the pandemic affected the entire planet. If we have everyone pooling all their, even people too, like manpower, we have everyone in the whole planet all putting all their money and manpower towards things, I'm sure we'd find other breakthroughs and things. It's just everyone was focused on this. That's kind of why it happened so fast. The world is filled with bright people. I'm sure there's so many other things we could have solved, right? But
Yeah, and to add to that, it was the example of when the mRNA was being tested in Zika virus, right? There were setbacks because of the funding.
yeah,
Now all this pandemic and everything kind of pushed everything, and like, okay, let's actually start testing and start doing this because we have all this data and all this testing. It didn't came out of nowhere. We have all this knowledge about mRNA, right? I told you it's a process that our body uses to form proteins, right? And they had all these tests that were done before, but they were just stopped there because of the lack of funding.
I also want to add
And
viruses and have been studied for so long. We've made so much progress within the past 20 years, I think, Professor Jangra said in class. So we have all these years of collected information that kind of helped out with making this new mRNA virus, because we didn't have to start from scratch when we had such a good foundation of information.
also scientifically, I don't want to use the word that we got lucky, but in some ways, yes, because if this virus was something like HIV, something like influenza, something like hepatitis C virus, where the field is struggling to generate a vaccine for a long time, because those viruses are very different. With this one, we got lucky because we took the spike protein as our target vaccine, and it just worked. It just like more than 90% protection. So in some ways, it's good that we had a coronavirus, that we knew something about them, and we were able to get to the vaccine in 1 year. Initially when it was proposed, in 3-4 months of this, all the virologists were like very skeptical that is it going to work? What if we are putting all our eggs in this one basket, which is dependent on this one protein? But it amazingly worked. So we got lucky that way.
so we got a question about pregnancy. Can you get the vaccine even though you are pregnant? Does it affect the embryo? So I actually did look into this a little bit because my sister's pregnant. So when I was doing all the research for this, I was looking into it. So basically, I don't want to tell you to get the vaccine because you definitely want to talk to your doctor still. But basically, it's safe for pregnant women. There was actually a study released last month, like the 21st or something, and it was only like they had certain rates, but it was the same amount of rates as unvaccinated people. So right, if there was like 2% stillborn or something, that was the same rate as if you just took people that were unvaccinated. So there was no actual statistical difference. There might be some thing there, but because there's not a crazy amount of research on it, right? They're always just going to research regular plain Jane people. They're not going to do the outlier groups yet. I mean, I'm sure they're doing the research now, right? Like this was only published last month, but I would definitely still talk to the doctor, 100%.
There was also, oh, sorry, Brian, go ahead.
Well, I was just gonna say, I know my sister said she was gonna get it. She just said, screw it, I'll wait until I have a baby and then I'm gonna get it. But just.
There was a follow-up question that said that somebody was pregnant and had COVID, and she was wondering if she should get vaccinated to protect her baby. And I think, again, that's a personal choice, but I think if you want to look at it kind of from an objective point of view, if you had COVID really early on in the pandemic, it's possible that your antibody levels have declined over time. And again, as Rohit said, most of these antibody titers that they've analyzed are still protective. But if you are worried about potentially being reinfected or contaminating somebody else, getting the vaccinated would kind of act as a booster shot. So it would just boost your immune system and increase those antibody levels to higher levels again. And then I think somebody said that women have been showing stronger side effects compared to men. Could this have to do with their stronger immune response, or could it have something to do with dosing, which I think is really interesting, because if you've gotten vaccinated, you've probably noticed that there isn't a difference in the dose that men and women get. And yeah, I mean, I think that for a lot of medications, dosing for women in general tends to be slightly problematic because it's not taken into account quite as much. But I also think that for manufacturing purposes, this probably really expedited the production.
Also, the thing is, the way rules and regulations are set for these trials, and they are set for the right reasons. You cannot straight away go into kids and test it. You cannot straight away go into pregnant women and test it. You cannot really use a particular group of people to just test a vaccine. So you cannot just, say like, we're going to test this vaccine straight away in only women or only men. And those rules are in place to protect these protected groups and every one of us. So to really have to, and this goes back to all these questions about why did we go with this particular dose? Why did we go with 2 shots versus 1 shot? Is because that is what we tested and that is what we know works. We cannot just go and change these things according to our will, because then that would mean we have not really tested it that way. And where do you stop? So the stop right now is, where what we tested, we stay with that, and we don't change things just according to our wish. And
A question I saw earlier was, how does COVID differ from the flu? Which is a great question, especially since personally, we're going into allergy season, and I don't know if you guys can hear me, I'm a bit affected by that. But COVID and the flu, they have a lot of similar symptoms. For COVID, the most, the most prevalent symptom that lets people know that they have COVID is the loss of taste and the loss of smell. That's the most distinguishing feature. What I did when I had the flu earlier or late last year is I went and I got tested. There's a lot of places out to get tested. There is, it's better to be safe than sorry, just because there's so many similarities between the two.
I also think that as new research emerges, there have been some more severe long-term effects that have been associated with COVID. You've probably heard a little bit about brain fog with certain individuals. Some people have had digestive issues. So there's definitely research that needs to be conducted to validate this, but it does seem like COVID or a SARS-CoV-2 infection can have longer term consequences that haven't necessarily been associated with other viral diseases.
And even there is some data that people who had long COVID, and that's just, well, anecdote, I would say. The data is coming, is that people who have had long COVID, when they get vaccinated, some of them are seeing those long COVID symptoms go away. So that's another interesting thing that we don't understand how that's happening, but it's very interesting. There's another question about if someone presents with moderate to high antibodies, do you suggest they get vaccinated sooner than later? I think people should follow what the recommendations are. And, as Vinita was saying earlier, that we still need to follow what the doctor, we need to go to the doctor and get the vaccine and follow the recommendations, what the doctor says. So, what we are presenting is a collective picture where the data tells you we are not, MDs, giving you prescriptions. So please be careful about that. But I think it makes sense to stay in touch with your doctor and ask.
So do we know? There's a question about does COVID-19 virus? We normally believe it infects the lung, but does it infect anywhere else? Do we know? Because that also follow up with the long COVID, right?
Yeah. So we have all heard of like loss of sense of smell and taste, right? So that is related to it infecting some of the neurons that are involved in detecting those senses. So it's not just lung tissue; it infects a lot of other tissues too, and that's why in some people we also see, one of the symptoms is diarrhea, right? So it's not just respiratory symptom. So yeah, it's not just limited to lungs.
Great.
We have ACE2 receptors, right, on a lot of your organs, right?
Exactly.
Yeah.
This is the first time, and it's very exciting that we have such a large audience, and we run 30 minutes over time, and that we planned it. The students you already know, and you can reach them for additional questions, or you can email me. I'll be happy to relay all the questions. Now, this event has been recorded, and the recording will be available either through biology department or through the Alumni Association. And we all appreciate everyone's interest, and this is the current challenge that we have. And I think hopefully you can learn and help all three students spread their effort to help our young people, especially the younger generation, to better understand the vaccine and the pros and cons. It is a personal choice, but we're not joking, but it's statistics, right? And you have to consider what those people who are around you and who would you affect if you get the virus. And yes, you may you may have to suffer a little bit, or you worry this, you worry that, but you have to balance. So and I like to thank all the speakers, the three students, for the excellent talk, and I like to thank three professors for guiding these three teams for producing these three professional talks, and I like to thank everyone for spending time on a Saturday morning to support our students. And I hope this is the beginning of the sign of the Lehman faculty and student and alumni being one big family, helping everyone stay healthy and going forward together. So thank you all. And yes.
Dr. Chang, can I make a final comment?
Yes, please.
Whether you want to get vaccinated, or your want to take your family and convince your friends and family for vaccination, there are two ways to do it. One is to use data, and this is what we talked about. The other way is, as humans, we are really driven by anecdotes. And if this makes you believe, I have a 1-year-old kid, the day he is eligible to get vaccine, I will get him vaccinated. All my family members have been vaccinated, whoever had a chance to get it. So, please use whatever conviction you need to get vaccinated, but, please try to, make a sensible decision, whether it's data driven or it's driven by out of love for your, parents and cared ones or whoever, but please. Thank you. And thanks to, especially it's been, we are, as the three professors who really worked with the teams and presenters, Sasha, Vinita, and Brian, we're really proud of you, the way you presented and you did it. Thank you so much. And thanks, Dr. Chang, and everyone for giving us the opportunity to do this. And it's great. Thank you. Sorry for interrupting. I needed to say that.
Thank you. Thank you very much. And enjoy the beautiful weekend. And for the students, best of luck for your finals. And most of all, everyone stay healthy. Thank you.
Thanks, everyone.
Bye.
Bye.
Good job all around.
Thank you.
Thank you for all the help.
Very nice.
Take care, guys.